the HCA Equity Working Group co-chair, and who is based at MIT and the Broad and the Reagan Institute. So Alex, welcome, and please, over to you.
Thanks, Christine, and welcome everybody. Thanks for joining us this morning. I'm going to kick it off by providing just sort of a welcome and a brief overview of the HCA. I'm probably the least exciting part of this presentation because the real goal of this day and the next day is to really ground you in the experimental and computational pieces necessary to do some of this work yourself. But I wanted to give you sort of a broader overview of the HCA. And if you're not already a member, we would love if you join. There's no fees, no blocks. You literally have to sign yourself up. And if you're having trouble, you can ask Christine. Shut my dog in so my 5-year-old doesn't get woken up by pot of the head. Apologies. If at some point it sounds like I'm talking to a 5-year-old, it's because my 5-year-old has woken up and is looking for me. Welcome to the meeting. As an introduction, just sort of what is the HCA or the Human Cell Atlas? Well, it's a broad community of people. It's a grassroots movement of individuals that are really interested fundamentally in creating a comprehensive reference map of the types and properties of all human cells as a way of understanding, diagnosing, monitoring, and treating health and disease. This is, for me, a nice statement, but a little harder to conceptualize than something like this. Basically, the goal is to create a periodic table of our cells. And being trained myself as a chemist and physicist originally, the idea that we would want to have sort of that same fundamental reference that's the basis for everything we do in chemistry or physics, but for biology makes all the sense in the world. And so you can imagine that the goal is to understand what are the different cell types that exist, what are the fundamental properties that differentiate them from one another, what do they look like in multiple different tissues, and to understand them at multiple different layers in terms of not only what they think at a given moment, but also things like how they're regulated and how they change in different disease states. So the HCA is a relatively new organization. So if you're just starting to get involved right now, don't worry, you're not late to the party. This is something that really began with a kickoff meeting in 2016, building off some of the work that had been done in the past, several years that had really developed some of these technologies where it was clear that there was a new way of studying biology at single cell resolution. And the question then became, should we as a community try and come together to build a reference atlas? And so quickly things have expanded, moving from a place where things like the analysis working group began to developing a white paper to working through things like standards in technology and ethics, to the formal initiation of the equity working group, which I co-lead with Partha and Musa. Now, I know that it looks like these things came years afterwards, but to be clear, all of these topics, including equity, ethics, and standards, were part of the original formulation. It's just in some of these, it takes a little bit more time to have conversations with stakeholders and think about what it is that makes the most sense. Specific initiatives began to think about pediatric networks. We built out the bio networks. COVID unfortunately happened to all of us, but it led to a place where there was a real impetus to use these data sets to try and create helpful references for people across the community. And so, along the way, there was a series of different meetings. The meetings were usually for people that were already pretty deeply invested to help coordinate and organize, but increasingly what we try to do specifically in the equity working group is to think about equity in action through having meetings like this that help to bring some of the knowledge and skill sets to individuals that are interested in getting involved. And so, there have been general meetings, regional meetings, originally in HCA Africa, ones that started through the equity working group in Brazil that eventually led to the formation of HCA Latin America. As well as things like equity meetings, which feel like they were recent but happened a little while ago now. To highlight sort of the growth of this community, Tracy's put together this wonderful graphic that sort of shows you where people have come into the consortium, how it's been built up. So it was something that was really dominated by Europe and the US back in 2018. There were obviously multiple different countries involved. And many members across institutes. But what we've seen as we moved into 2019 and then 2020 is the growth of involvement across multiple different continents and countries. So we move into 2021. What's nice is that you see an increasing representation from Latin America and from the Middle East and Africa, without sort of explosive growth here. So it is increasingly becoming, a global effort that involves people around the world. And part of this has been facilitated by the formation of networks. One of the first networks to form was HCA Asia, and so there's broad representation among the scientists within the region where they organized local symposia. Actually, there was just an HCA Asia meeting in Bangkok. That was followed by the formation of HCA Latin America after a meeting in Brazil, and that's really catalyzed a lot of initiatives and conversations locally. And more recently, the HCA Africa Regional Network's been formed, and hopefully will continue to grow in sort of a similar fashion. Now, in terms of how the HCA is organized, naturally it ended up being organized by tissues because that's where people had expertise who were working. And so I've listed this here, and I wanted it to be in the video so that you know who was working on, say, liver or eye or gut. So you knew who to contact and what the email addresses were in case you wanted to learn more about these specific areas. So just to give you an idea of some of the stuff that's been created, we're now over 100 million cells across, over 100,000 samples that spanned, almost 10,000 individuals. And so, a large number of these were collected during COVID and were very helpful for thinking about what was happening in severe and mild disease. But, to think about this from sort of an individual tissue level, if you're interested in, for example, the intestine, there's over 5 million cells across many samples, and so on and so forth. Similarly, there are other tissues where there's pretty extensive sampling at this point. So just to give you a little bit of background on some of the working groups that might be relevant to some of the things that you're going to do, if you have questions about technology or about how to pull them in or about what works best, any of us are happy to try and give you our own thoughts, but there is actually an entire standards and technology working group led by Holger or Rita and Xiaowei that really thinks about different methods and how to benchmark them and how to develop guidelines for their applicability. And so you can always ping them. Their emails are on the website. There's sort of a group email list for them that can help you think about where anything falls. Or if you're thinking about the need for developing new technology, you could always have a conversation with them. They might be able to point you to somebody that works in the space. Now, today and tomorrow there's going to be experimental work and discussions of that, but there's also going to be computational work. And so not only is there a technology and standards working group, there's also an analysis working group led by Donna and John. And this really brings together people that are interested in computational work and focuses on outstanding problems through jamborees or sort of hackathons, however you like to think about them. And one of the big things that they do is not only think about how you should computationally query this, but think a lot about how you can create sort of a metadata or a standard for describing everything, because you need sort of agreed upon ways of referencing different cell types. Another working group that I'm particularly passionate about and I co-lead with Liz and Partha is the equity working group. And this is really focused on trying to push equity in the HCA. When people think about equity, they very often think about equity with respect to representation. And we think representation is important. We should have references that are relevant to all of humanity. But I'm a big believer that we can't have representation without participation, thinking about benefit. It's important to engage people around the world in creating these references towards creating the right references, understanding regional barriers, doing things in ways that really respect the communities with which you're working. And then, really thinking about how to make sure the benefit from this doesn't disproportionately accrue to certain groups, but really helps to be beneficial for people all around the world. And so what we focused on to try and get to this idea of participation and benefit is to really try and enable people to participate through outreach, education, training like we're doing today. And so, our real focus has been on how to engage more, trying to think about metrics and monitoring them because, things that matter get tracked and measured. And they're not always great when you look at them at first, but it gives you an idea of where to grow. The other thing that we're acutely aware of is sort of issues with funding. And so we try and work with funders. HCA is a grassroots organization. We don't have money ourselves, but we are happy to go and try and encourage individuals at different funding agencies to focus on increasing funding for recruitment and training. And actually the workshop today is the result of success there, where we've got some support to do it. And the other thing that we do is a lot of training workshops, some of them in person, like you can see here, this one in Brazil, where Moose and I went down, or the meeting in Addis. But, Hopefully more of those will happen in the future in person. And I know we have plans to be in Ghana in person in the spring, as well as Nigeria next summer. So in parallel with the equity working group, there is an ethics working group. Equity and ethics are related, but also distinct. This is led by Bartha, Barbara, and Roderick. And really it focuses more on sort of the legal and ethical issues that show up as you think across geographies and think about sort of the creation of some of these references. And they've helped build things like an ethics toolkit and think a lot about, what consent is, how you obtain it, what are the rights of individuals. And they're doing very important work. If you're interested in that space and sort of the policy behind this, I would strongly encourage you to speak with them. I wanted to highlight really quickly how you can get involved in making your work part of the HCA. And as I said, it's a grassroots movement. You can sign up, you can go online, you can become a member at any moment, and I encourage you to do so. But you can also make sure that all of your publications become part of this. And there's a specific procedure to follow. You just drop an email, fill out a submission form, add a specific acknowledgement, and then let the HCA know. One nice thing about this is you might say, what do I get from it? Well, the HCA does try and publicize and promote some of these things. So it's a good way of drawing additional attention to your work, which, hopefully is beneficial to you and your trainees, or to you if you are a trainee. So the HCA at its core is really focused on creating this healthy human cell atlas, but many of us do not just think about health, we think about disease as well. And I recognize that in many parts of the world, the idea of creating a healthy reference without a disease reference is a luxury. And so to me, they go hand in hand. And the real reason to create the healthy reference is to understand human disease, to understand what differences are, to understand what you should be targeting. And so whereas the Human Cell Atlas focuses on thinking about the healthy reference, in parallel, there are a number of initiatives around the world that are really focused on trying to understand disease, whether it's the Human Tumor Atlas Network, some specific things like Lifetime in Europe or so on. And so we think about them going hand in hand. And very often, for my work, which focuses a little bit more on global health and infectious diseases, I think about trying to not only create a disease atlas, but trying to create the healthy reference as a basis for understanding what's changed, because the biology can be a little different in different regions of the world or in different communities. And so with that, I'll just welcome you once again to the HCA and thank you all for joining us this morning. We're really excited that you're part of the community and that you're here with us. I'm happy to take any questions you might have. Maybe you have questions now, maybe you don't, but you can also email me at any time. It's just my last name at mit.edu. I'm happy to introduce you to anybody in the network. I'm happy to have conversations about anything that might be on your mind. And we're delighted that you're here. We do hope to hear from you. We really do want to engage a broader swath of individuals at multiple different stages of training in the HCA. And so there's anything that we can do to make this, a better community for you, please let us know, because that's why we're here. So with that, I'll pop out and say thank you, Christine, for dropping my email in chat.
Thank you, Alex. Yes, so Alex's email is in the chat, so feel free to reach out to him at any time. If there are any questions in the audience, feel free just to unmute yourself and ask directly, or you can post questions in the chat and we can ask on your behalf. So I'll give it a minute or two. Okay, so maybe we'll move on to our next speaker, Daniela Russo from the Shalek Lab. Daniela is a research technician, and she is here to introduce you to experimental sequencing methods. So Daniela, welcome, and I'll send it over to you now.
Thank you. Hi, everyone. Welcome to the first lecture of this series. So like Christine has just mentioned, I'm going to discuss some experimental sequencing methods. So for today, our overall goals are going to just first and foremost introduce to this rapidly expanding world of single cell transcriptomics and really try to focus less on specific tools, but more on the underlying concepts. And this is to give everyone an opportunity to understand the whole breadth or a good chunk of the breadth of the field, and then later make informed decisions based on that information. Hands-on exercises are going to help us to get, an understanding of single cell heterogeneity and just some of the first steps into tackling this sort of data. And most importantly, we want to just have some fun. We want to learn, we want to collaborate and really form an interactive environment with everyone. So please feel free to ask questions as needed, interact when you can, and all of that good stuff. The expectation for this course is, or for all of this type of virtual event that we are having, is, if possible, just to raise your hand if you have a question. As I mentioned, I'm always glad, and all of the leaders are always glad to answer questions. It's just helpful if we can identify that by raising a hand in the chat. And if not asking a question, just to keep muted so that it's a little bit easier to hear whichever training leader is speaking at the time. And if we run out of time, please always feel that there's still time for questions. So we're going to use a Slack channel, and that is a great place to ask questions after a lecture or even after this whole training is complete. We always want to be there to help you answer questions. So please feel free to use that resource. And then with Zoom and all of these technologies, just please be patient if there are technical issues. We're going to do our best. The team, Elliot, Christine, and Tracy are incredibly exceptional and are always working to make sure that this runs smoothly. As I mentioned, we're going to use Slack for a lot of post-training questions and any kind of assistance that you might need in the interim. And so if you haven't been introduced to Slack previously, it is an application that you can download if you go to slack.com, and typically it is just dependent on your operating system. So there's a Windows link, a Mac link, etc. We should have all received an invite to the HCA Slack channel. It would look a little bit as you see on my screen now. And it really just follows a kind of, there's options for chat. So if you Google someone's name, you can Google my name and you can not Google, sorry, but you can enter into the search bar my name and you can send me a message directly. Or there are these channels, as we see, the one here is the HC Africa attendees channel, and that would send a message or have a forum type discussion that includes all of the participants of this channel. So please, if you have questions, feel free to let us know in the moment, but also feel free to put them here. And so on to the good stuff. The first and foremost place that we'd like to start is with the cell itself. And so the cell is a fundamental unit of tissue, and diversity of this tissue is driven by some features. So the basic feature is the type of cell that we're discussing, for example, a hemopoietic stem cell or a T cell. And so if we think of T cells as the example, we consider the cell's location. Is this T cell circulating the bloodstream? Is it at the germinal center interacting with a B cell or other cells? And we also think about what state a cell is in. Is it naive and hasn't seen antigen yet, or is it a memory cell? And finally, we can take a look at what are cell divisions and transcriptional changes that are leading from a hemopoietic stem cell to a T cell. Oops, sorry. There we go. Single-cell genomics gives us insight into many cellular features, and comprehensively profiling the molecular processes of cells can help us to differentiate and define healthy and diseased behaviors. While several methods have been developed to characterize the different aspects of single cells, such as cell surface proteins with FACS or chromatin with ChIP-seq, there has been a significant focus on mRNA since it's relatively easy to capture and can be used to explore everything from everything that a cell aspires to be. Analyzing the transcriptome can reveal important information about the genome. It can also give us some insight into cell types and cell states, as I mentioned, and in turn, characterizing these distinct cell types can elucidate cell populations and cell-to-cell interactions that may be at play. As a result, several methods have been created in the single-cell RNA sequencing space to help develop and profile cells. Each of these methods has a specific focus, strength, and weakness, and we can see really the breadth of this rapid evolution, and that there's been some incredible growth in the field of single-cell RNA sequencing since 2009 and until now. And so we can see that there's been some introductions from manual processing, multiplexing, all the way to in situ barcoding, which allows us to look at more cells at one time. The main differentiation in this state, or before the emergence of single-cell RNA, is that we think that we know that RNA was profiled using bulk RNA sequencing, and this provides more of an average expression profile. The introduction of single-cell RNA allows us to break down this spectrum of signals by enabling the dissection of gene expression at the single-cell level. And so ultimately, bulk RNA will give us a signal, but single cell can provide further information on where that signal might be coming from. It can reveal very complex and rare cell populations, uncover regulatory relationships, and track the trajectories of distinct lineages in development. And this really helps in studying the molecular dynamics of different cell types during developmental stages or disease stages. And a more precise understanding is essential for elucidating the role and understanding of these beneficial or harmful states. We also know that heterogeneity is always present, and that a understanding of each of the cell types will help in revealing the changes of cell fate and mapping of trajectories. There is a general workflow that many of these single-cell RNA sequencing methods follow, and it is beginning with a heterogeneous sample in which DNA, protein, cellular debris is removed, and this helps to isolate the mRNA. This mRNA is then reverse transcribed and barcoded, resulting in a cDNA library. The library can then be sequenced, and we are expecting, or we are hoping, to receive millions of reads that will then be translated to genes by mapping to a reference genome. The final result is an expression matrix of cells and genes that are then further analyzed, with the main point or the main difference being how this barcoded library is generated. And these are just some of the main or different groupings of single-cell RNA sequencing methods. So here we've included SmartSeq2, DropSeq, InDrop, 10x Chromium platform, and SeqWell. And these different methods are often categorized based on their isolation methods. So SmartSeq2 focuses more on like some fact sorting into a 96-well plate, while DropSeq, InDrop, and Chromium focused more on droplet-based methods of capture. And finally, SeqWell looks at or uses nanowells to capture. Only the plate-based SmartSeq2 method can offer full-length gene coverage. And so when we think of this full transcript assay, we know that it has about 80% capture efficiency, which is very good. It's one of the strengths is that we can get about 80% of the cells back in terms of utilization for our analyses, and we are getting the full length transcript. It does tend to be a little bit more expensive per cell and is quite labor intensive and slow. And in this method, we have a lot of benefits in terms of the data in itself, but it becomes much more difficult to process anything more than 100 or maybe thousands of cells. The process between SmartSeq2 and SmartSeq3, which is a newer addition to this type of method, are quite similar. And so SmartSeq2 has really been around since about 2014, and just a few years ago, we have also seen the introduction of SmartSeq3. I'm going to highlight them in unison and then make the distinction from them a little bit later on. And so both of these methods are going to begin with a reverse transcription in which cells are lysed in a solution that contains free DNTPs and tailed oligo-DT nucleotides. And these are responsible for priming the reverse transcription reaction on the polyadenylated RNA sequences. These free DNTPs are added in the initial step to later improve the yield of the RT-PCR, or the reverse transcription PCRs, probably through mechanisms that stabilize RNA primer hybridization. There's also a template switching oligo, which has locked nucleic acid at the TSO3' end. And the big difference between these two methods comes in this TSO, or this template switching oligo, in the way that SmartSeq3 is using a template switching oligo that also has a TN5 motif, a 11 base pair tag sequence, and then an 8 base pair unique molecular identifier sequence. And we're going to talk a little bit more about these unique molecular identifiers a little bit later on. We then move to a full cDNA amplification using PCR, and this is consistent within both of these technologies. And then we are going to perform a tegmentation, and this is the instance in which a very similar TN5 transposase is introduced into the SmartSeq2 reaction. As I mentioned, in SmartSeq3, this happens more towards that reverse transcription step, but in SmartSeq2, we have this TN5 transposase that catalyzes the in vitro integration of these oligonucleotides. Finally, we're ready to sequence, and then we are ready to analyze after. And again, the big difference is at this step where the output of sequencing in SmartSeq2 is going to be these full-length transcripts. And we're still going to maintain these full-length transcripts in SmartSeq3, but we're also going to have the addition of this 5' unique molecular identifier. And what these unique molecular identifiers are these short random barcodes that we add to the transcripts during the reverse transcription stage. And the complex indices added to the sequencing library enable some accurate bioinformatic identification of PCR duplicates down the line, and are mainly to reduce amplification noise by allowing the deduplication of fragments. So basically, a little bit further into this process is that we will label mRNA molecules very early with these random nucleotides. And so we can correct PCR bias, but we aren't necessarily going to capture or correct for some low capture rates. And so it's good to make that distinction and to utilize these UMIs, or unique molecular identifiers, for their abilities and their strengths. The next method is a droplet isolation method, and allows for higher throughput than the SmartSeq2 or SmartSeq3 platforms. Many of the current high throughput single cell sequencing methods are droplet based. And so, for example, DropSeq uses a microfluidic device, and individual cells and microparticles are responsible for delivering these barcoded primers and are loaded at a low density to randomly encapsulate your cells. After lysis and hybridization, you obtain a set of beads that are called single cell transcriptomes attached to microparticles, or termed stamps. And these stamps can be subsequently reverse transcribed and amplified and pooled for sequencing. InDrop expands on this technology and instead uses hydrogel beads. And then finally, we also have the 10x chromium set, which uses gel-like beads that contain these barcodes and can dissolve in the droplet. While all three of these workflows generate droplets, we can see here that they each have their own strengths and advantages or disadvantages. And as discussed before, these droplet-based methods also present some differences in terms of scalability or capture, as we mentioned with SmartSeq, which had an 80% capture rate. And DropSeq tends to have the disadvantage of this low capture efficiency. The result is that we have this emulsion happening and you end up with lots of droplets, but only very few of these droplets actually encapsulate a cell. Another common issue is in terms of scalability. And so we have a very difficult time in having this happen very synchronously between samples. And when you have extremely rare samples, you don't want to waste time. And so sometimes, or, take a long time. And so sometimes these aren't going to be ideal due to this low capture efficiency. We have 10x, which actually does scale well and has better capture efficiency. But as you increase the number of samples, this tends to be an incredibly costly option. And so sometimes we are trying to really balance a cost-effective method with a capture efficiency. And so in many cases, 10x reaches that capture efficiency, but might have some struggles in terms of cost effectiveness. To address this issue, we can think about SQL as a solution.
You want to?
And so due to low efficiency in encapsulation, we can, instead of using these barcoded beads and droplets, SQL has used an array that has sets of little boxes, and these act as compartments for beads and cells and enable us to do a lot of aspects with gravity. And this makes it more efficient and easier to implement. We call these little boxes sub-nanoliter wells, and the purpose is that they can really accommodate one bead into their well. And this allows for a really high single bead loading efficiency, which is at about 95%. Cells are also gravity loaded, and due to the size of these sub-nanoliter particles also helps increase the capture efficiency to a rate of about 80%. Another advantage of SQL is that it is also very portable. So with an array, a pipette, a clamp, an oven, and a tube rotator, you can really implement this technology in a variety of places. At a high level, some of the steps for SQL are very similar to other single-cell RNA sequencing protocols, but I just want to quickly touch upon some of the aspects that make this unique. And so with SQL, cells are extracted and dissociated. And instead of loading the beads and the cells into something like a 10x device, we load them onto these arrays. We then conduct a cell lysis, and this is done using a permeable polycarbonate membrane. So this key feature of SQL allows for the cell lysis and facilitates buffers to be exchanged while blocking other biological macromolecules. And so this is done to improve transcript capture, and it also reduces cross-contamination. Once cells have been lysed, we have a bead-bound poly DT oligonucleotide, and this will also have this primer sequence, cell barcode, and that unique molecular identifier that I was mentioning earlier. Next, we peel off the membrane and we remove the beads, and this will be done to allow for subsequent bulk reverse transcription, amplification, library preparation, and then ultimately some sequencing. And so to just think about some of these in a little bit further detail, that reverse transcription step, we think about as that once the beads are with the hybridized mRNA have been isolated, this reverse transcription can occur to generate full-length cDNA. So we incubate a reverse transcription mixture that has, again, another template switching oligo, and thus will hybridate, this oligo will hybridize to the 5' untemplated C bases and allow for this cDNA synthesis to occur. In order to remove any of the excess primer, we will then subject this cDNA to an exonuclease treatment. And that will really end this reverse transcription stage and take us to this second strand synthesis. So during the reverse transcription, sometimes we don't get this template switching oligo, and it's really essential for amplification. And so once we have finished our reverse transcription, we are trying to improve transcript recovery. And so we will conduct this second strand synthesis to incorporate a random primer that we term DN Smart, which will bind to parts of the cDNA to recover transcripts that would occur as a result of inefficient template switching. And so to do that, we perform denaturation by washing the barcoded mRNA beads. And with the help of a clino enzyme labeled with the SMART sequence, there will be primers that first let us make full-length cDNA, but will also then come down and bind to its reverse complement. So here we're just showing one strand. And then from there, we would have our double-stranded DNA, and we would take that product and amplify using PCR. And then we would isolate and clean this product using 2 rounds of spry purification using Appiah spry beads at a 0.6 and 0.8 volumetric ratio, respectively. We then quantify our product using a qubit fluorometer to measure concentration before prepping the sequencing library. To make this library, we will tag randomly with Nextera barcodes. And so during tagmentation, the DNA gets cleaved into random fragments. However, only the 3' end of the cDNA fragment will be tagged with an S7 sequence, a universal Illumina primer, and then amplified for use as a library. The P7 primer anneals to the 3' end and adds a P5 primer, which is going to then anneal to the 5' end. And the transpose adds these adapters randomly, which are essential for Illumina sequencing. The final library product is then cleaned again via a spry cleanup method. We are then going to pool and quality control before we load this library onto our sequencer. This data can then be demultiplexed, quality controlled, and as we show here, a t-SNE plot, which is a common analysis tool for single-cell RNA-seq data, can be generated. Oh, perfect. So overall, there are many different types of single-cell RNA sequencing methods, and this is just another figure showing the same sequencing methods that we've mentioned previously, but really highlighting that each protocol has advantages and limitations. What ends up being one of the major differentiating factors between which method we are going to select is first of all, the biological context in which we are selecting a method, the cost of a method, the objective, which is to say the output of these methods. And overall, it is really important for us to make considerations. And so some considerations for single-cell RNA sequencing start with choosing the right protocol. And so as I mentioned, we want to think about our throughput, which is the number of cells that we want to process overall. Or is it hundreds? Is it thousands? Is it tens of thousands? We also want to think about the sample of origin. If it's frozen or fresh will depend on the success of certain methods. Cost and labor and time limitations should also be considered, as well as gene body coverage. So do we want more of a 5' bias, a 3' bias, or are we most interested in full-length transcripts? Then we want to think if the addition of a unique molecular identifier is going to be helpful, and to what degree that will be helpful, as well as the sequencing depth or the sequencing depth of a cell specifically. So for example, if I wanted to classify all cell types in a diverse tissue, I would make sure that I'm using a more high throughput system or high throughput method. If I want to re-annotate the transcriptome, I want to discover new isoforms, I'm more concerned about having full-length coverage. And if I only have access to an archival human sample, I'm going to want to choose a method that permits fixed cells or nuclei processing. And each instance is going to have its own set of considerations and challenges that are going to be involved. But ultimately, there are lots of options and lots of opportunities to explore this field. And with that being said, I'm happy to take any questions related to single cell RNA sequencing methods.
Thanks, Daniela. So as we said before, we're all friends here, so feel free to just unmute yourself and ask questions directly, or you are more than welcome to put questions in the chat and we can read them off on your behalf. But I think we'll give it a minute or two, Danielle, to see if questions start rolling in before going to the next thing on the agenda. Okay, I don't see questions coming in. So what I suggest that we do is we have a 15-minute break. We're running early, so that's good. So why don't we restart in 20 minutes? So that's going to be one moment. It's going to be 8:05 a.m. Eastern time. And that will be 1:05 GMT time. So please go get some coffee or tea, and we'll see you all back here in 20 minutes. Thanks, everyone. Yes, so Collins, it's over to you now. So Collins, you are a researcher at the at WACBIB and the University of Ghana, and you'll be introducing the computational analysis methods to us here today. So over to you.
Thank you, Christine. Welcome everyone to this workshop where we are going to talk about, single cell methods. And I'm going to basically focus on giving a small introduction on the computational analysis methods that are involved in single cell analysis. So we form part of the Human Cell Atlas Africa Network. And if you like to join and you're from Africa, you can always reach out to us and we'll guide you on how to do that. So just to start off is that, after the sequencing that has been talked about by Daniela and you've obtained your data. So basically now we are asking the question, what cell types? How can we identify the cell types? What do you do after your samples have been sequenced and you've obtained your single cell data? So you can think of, having all different cell types and a stem cell parental advice whereby each cell is being told you can be anything that you want to be when you grow up. So once you take all these cell types, you sequence them, they're all mixed up. You need to be able to separate and identify that this is this particular cell type, this is this particular cell type. So the Human Cell Atlas, I think they aim to construct population level maps for human phenotypic variations and also illustrate single cell profile of an individual based on their phenotypic phenotype. And you can think of it like, all these small boxes being different cells. And if you put them on a like a 2D space, you're going to have different cell types clustering according to their phenotype. And you can see that, for example, this represents population level variation, but then you can see only a few cells that are representing healthy states. And then you have a lot more cells representing disease state A, and then disease state B, and the cells that are representing pre-disease state. So you can see that because all these cells express different genes, they kind of tend, if you put them on a 2-dimensional space, they will be able to separate according to the genes they're expressing. And disease A is probably driving a particular cell expression of particular genes. Disease B is driving expression of particular genes. So because of these differences between cell expressions, you are going to have cells separating on a 2-dimensional space. So basically, this is what we use. We try to form clusters from the single cell data so that we can see our cells separating according to a particular phenotype and what could be the particular phenotype or particular different cell type. So this is basically what we do. Now, I know most of you probably have done sequencing, and from sequencing you get reads. So what you do is that, I think Daniela has talked about this, that there are unique molecular identifiers, and then you have cell barcodes, and then you have your sequencing reads. So the first thing you do, I hope you can see my cursor. Yeah, so the first thing you do is that you take all these reads in their millions. Normally from sequencing, you get almost 200 to 400 million reads. Now, the first thing you do is you do demultiplexing. And this is whereby because you can see there's color coding here for cell barcode, you can see green, green, green. The UMI too, you can see 2 that are blue and that. So the first thing you're going to do is you're going to separate the cells as per the cell barcode, and you're going to take all these mixture of reads and separate them to cell-specific reads, whereby you have each particular barcode separating to its specific cell type. Now, once you do this, you do mapping, and this is whereby you map to your particular reference genome. And in this case, since we are talking about the human cell atlas, you're going to obtain the reference genome, and then you're going to map all these cell-specific reads to the reference genome. And this is basically how they're going to line up. So based on the sequence, similarity to the reference genome, you're going to have reads mapping to this in this format. And what you do is that the next step is to quantify, and this is whereby you take a particular gene region. So assuming this is a genome, you take a particular genome region, and then you see how many reads are uniquely mapping to that particular region. And you do this, thus quantifying the total number of reads that are mapping to that particular region. And then so you can be able to count. For gene 1, you have 10 genes. For gene 2, you have 20 genes. For gene 3, you have 30 genes. Now, you are also able, because you had cell-specific reads, you are also able to count for each particular cell based on its unique barcode, how many reads are mapping to it. And at the end of the day, what you obtain is a gene expression matrix that has cells, cell 1, cell 2, cell 3, all the way to the total number of cells. If you have 10,000 cells, you're going to have 10,000 cell types. And each cell type, you have each gene 1, gene 2, gene 3, gene 4, all the way to gene M having a specific number of reads. So you're going to have a more like an expression matrix that has this particular number of genes, more like a 2x2 table. Those who know what a 2x2 table is, whereby each square represents the total number of genes in that cell and the total number of reads for that gene in that particular cell, all the way to all the total number of cells that you have in there. Now, once you do this, once you obtain a gene expression matrix by counting all these reads that are mapping to that particular gene, the next step you're going to do is that you're going to do normalization, imputation, and feature selection. So what has been done is that some of these processes have been containerized into pipelines and have been containerized into pipelines. And some of these pipelines are called Cell Ranger pipeline, drop EST, and etc. There's a few others new being developed and all that. But mostly this is the main pipeline that is being used for some of these processes. And basically you don't have to do this manually yourself. So once you have your raw reads, you just put them on the Cell Ranger pipeline. And at the end of the day, what you get is a gene expression matrix. So the next steps that you do is that now you have to start thinking about what could be wrong with your data or what could be right with your data. And this is whereby you look at some of the technical consideration, and these include sequencing quality. What is your cell quality? What is the expression quality of your data? And also, is there any level of differential expression between different cell types or within a particular cell type and across phenotypes? So I'm going to break them down a little bit by a little bit and talk about the sequencing quality, expression quality, and gene expression. So, as I said, you have your cell types, you do single cell RNA sequencing, you get a gene expression matrix, which has the total number of reads that are mapping to a particular gene in a particular cell. So the first thing you do is you compare the percentage of unmapped reads and the percentage of reads that are mapped. And if you get that you're having a high percentage of reads that are being unmapped in millions compared to the number of mapped reads, then you might be having a poor quality cells. So what you want to have is that you should have a low percentage of unmapped reads. You should have a low percentage of unmapped reads, but a high percentage of mapped reads and in high quality cells. And this is one of the things that you want to see. So I've put some reference papers on the side in case you want to read more about, what some of these figures are talking about. You could note down and have a look at it later on. So the next thing you do is you should look at cell quality. And ambient RNA has been one of the biggest challenges in sequencing data. And sometimes you want to remove this ambient RNA. And one of the softwares that has been developed is called CellBender. CellBender removes ambient background and sometimes it removes backward swapping. So there could be a possibility that a particular cell type that has backward A has swapped its backward and is using, you are seeing cells sharing backwards. So you want to remove this, you want to remove the ambient RNA. And this software is was developed using artificial intelligence and uses deep learning algorithms to be able to remove this ambient RNA. So more like the data has been trained using a lot of ambient RNAs, and then now you can be able to use it to predict what RNAs in your samples could be ambient, and it will remove them. So it will look for transcripts that are being expressed in unexpected cell types and remove those genes from all subsequent analysis. For example, if you see hemoglobin genes being expressed in T cells, then you don't expect hemoglobin genes to be expressed in T cells, because hemoglobin genes should be expressed in red blood cells. And these are genes that are involved in, or proteins that are involved in carrying oxygen. So they are found in red blood cells. So if you find them in T cells, then something is wrong with the data, or that is more of ambient RNA or ambient background. So it's going to remove some of these genes that are not expected to be found in particular cell types. So typically, it will also check. So for example, if you make a plot whereby you're looking at mouse genes and human genes, you can see that this is the raw data. Ambient RNA will be more closer. If you are using human genes, then you expect them to be, farther away from mouse genes. So you could see ambient RNA, some that are swapping and some that are both. So you want to remove all this and only remain with genes that are uniquely mapping to the human genome. So the other thing you do is you look at cell quality, and one of the things is that you do normalization. So because each cell will be expressing or having a different gene expression profile, so you want to normalize for each cell so that they are all operating within the same range. I mean, I think normalization cuts across from statistics. Any data set that you have, you always want to do normalization so that you can have a good distribution of your data. And the reason why we do normalization is that some cells that have been sequenced tend to have different depths. So some cells could be having more reads being sequenced compared to other cell types. And some cells of different types could have different amounts of mRNA, which is a biological issue. For example, if you have monocytes and T cells, and you are seeing monocytes expressing more, let's say CXCL10, while T cells are expressing CXCL10s in a fold of 1, while monocytes are expressing in a fold of 10. Then it tells you that your cells, your monocytes, there could be just a biological problem. So you want to normalize so that the differences between these two different cell types is not so huge. And then sometimes you could have more highly expressed genes that are more variable. So you have, for example, the same gene that I talked about, CXCL10, having a very high expression in one monocyte, but very low expression in another monocyte. So what you want to do is you want to normalize across all these monocytes so that you have relatively even cells. So that's cell quality and normalization. What you want to see is you want to have a normal distribution and not distribution that is skewed to the left or to the right of the normal distribution plot. And sometimes what happens is that you could go through cycles. So you do normalization, you go on, analyze your data, you realize there's a problem, you come back, you do imputation, you move forward, then you remove some cell cycle cells, you do batch correction, you go back. So you could do, when you are assessing cell quality, is a process and is something you could do over and over through similar steps. adjusting different methods. For example, if you do log normalization, you realize it's not really correcting for everything. You want to go back and use a different normalization method. So some of the recommendations that have been made is that you should perform quality control by finding outlier peaks in different number of genes, the count depth, and the fraction of mitochondrial genes. So that's something that you should do. And you should also be permissive of QC thresholding as possible. And once you do your downstream analysis, even if you have done all the way to gene expression analysis, you should always be able to come back and redo and revisit your quality control steps or thresholds if the downstream clustering cannot be interpreted. So for example, you've gone on and you still can't find a specific cell type. For example, you did sequencing of PBMCs and you can't find T cells or monocytes within your data. You can't find a cluster within your data that has monocytes or that has T cells. So you realize that, okay, there could be a problem with my quality control. So you should be able to always go back, revisit your quality control, and come back and continue the analysis. Sometimes if the distribution of the quality control covariates differ between samples, QC threshold should be determined separately for each sample so that you can account for sample quality differences. Normalized data should always be, log X+1 transformed for use with downstream analysis method that assume that data is normally distributed. So whichever normalization method you're using, you should always try and use a method that accounts or assumes that the data is normally distributed for downstream analysis. So here are some of the key software that can be used in different stages. As I said, you can use Cell Ranger, Drop PST to do the whole pipeline analysis from reads to producing gene counts or gene expression matrix. STAR is one of the software that is used for mapping, and you can use STAR independently of the Cell Ranger or the Drop PST method if you already know how to do the other steps, especially generating the gene expression matrix. Now, doublets is one of the problems that is always found in single cell data, and I think Daniela talked about it. So this whereby you normally find, more than one cell type. You always find 2 cells that you expect to be expressing different genes, but they're expressing one gene. And these tend to cluster together because there's a kind of a confusion. They don't tend to cluster uniquely to a particular cell type. And this could be doublets. So sometimes you want to score them, and the good thing is that there's some softwares that have been developed to be able to remove these doublets, such as Scrobet and Doublet Finder. So you can use this to do that. Now, downstream analysis, and this is one of the softwares we are going to use subsequently in our workshop, is to use software like Surat, ScanPy, Scran, Pogada, to do downstream analysis from a gene expression matrix. And these are all free softwares that you can download. There are so many other softwares that you can find. You can go to this particular link here and find more softwares and tools for different stages that you can explore. So once you have your expression matrix, you also want to evaluate the expression quality, and you process the expression matrix. You remember I said your expression matrix, you normally have cells and genes, and inside here you have read counts. So the first thing you do is feature selection, and then you do dimensionality reduction. So this is whereby you can think of it like you have 10,000 genes on the axis. So you want to reduce them to smaller dimensions that can be, quickly analyzed or understood. And you can also estimate cell to cell distances. This is whereby you check what are the differences between cells. And you can also do k-nearest neighbors. This is more of the clustering step. So some people prefer to do k-means hierarchical clustering, while others now are using Liouville methods in graphical maps. So we are going to talk about this a little bit more in subsequent steps. So how do you do dimensional reduction? I think this is something that Or is going to talk more about in subsequent lectures. And because high dimensional data is very difficult to interpret, and one of the approaches to simplify this is to assume that data of interest lies within a lower dimensional space. And as I was saying, you could have 10,000 cells and 10,000 genes. What you want to do is reduce them to fewer principal components. This is more like compressing a certain number of genes into a particular component, and you have low dimensional data. So some of the common methods that are used to do dimensional reduction are principal component analysis, independent component analysis, multi-dimensional scaling, and non-negative matrix factorization. We might not be able to do all these methods, but at least we'll talk about the principal component analysis in subsequent steps. So the reason you want to reduce dimensionality of genes is that, take for example these two plots. When you have 500 genes, you are able to see this cluster clearly. You are able to see this cluster clearly, or at least separating clearly from the rest, from the other two clusters, and these clusters and this cluster. But when you have 20,000 genes, what you see is that there tend to be an overlap. You tend to have an overlap of genes across different clusters. And because of this, you cannot confidently say that this is cluster X and this is cluster Y. So that is what you want to be able to do, is that you want to reduce the dimensions so that you can have less genes forming a particular cluster. So it is recommended that you select between 1,000 to 500 highly variable genes depending on the data set complexity. So to be able to do clustering. Now, one of the methods that is being used these days is that we use graph-based clustering method that is nothing more than community detection. So instead of using, for example, k-means, which calculates the mean distance between two cells, you want to use community detection. And this is whereby you just put them on a two-dimensional space, calculate cell-to-cell distances, and then check which cells group together on a particular graph. And one of the methods that is being used is Liouville. This is applied on surats, and we are going to see it in action in subsequent sessions. So basically, there's no point of performing hierarchical clustering of 10,000 cells if 90% of the pairwise distances are null. And this is the reason is that, for example, if you have T cells, you could have 80% of the genes being expressed in different T cell subtypes being similar. So what is the point of using pairwise distances? Because the difference between those distances is null, or the distances between those subtypes is null. So you want to use a method that is able to cluster each of these T-cell subtypes as a close community of cell types that are close together. So one of the problems that we normally encounter in analyzing, and you have to be very careful once you do your dimensional reduction, is that you should be able to check for batch effects. And allow me to walk through this slowly. For example, if you have 3 biological groups, so you have group 1, group 2, group 3, represented by these icons. During processing for sequencing, you process batch 1, you process a particular batch as group 1, which is the same as group 1. So you process it separately. Group 2, you process it separately. Group 3, you process it separately. Now, at the end of the day, we are not going to be able to determine if the variation is driven by the biology or the batch effect, because if you end up with a principal component like this, whereby all the green, all the batch 1 samples are clustering together, you can go back and realize that, okay, batch 1, they all belong in group 1. And batch 2, batch 3, they all cluster uniquely separate. At a first instant, you might be excited. Oh yeah, you have very good data clustering separately, but that's not true. So what you want to do is that you want to have all these clusters separating as per unique cell type, not because of how you process them in different days. And this is how you want to be able to do it. For example, if you have group 1, you want to split the data into 2 replicates. Then you have group 2, you split it into 2 replicates, and then all these data, you process it, you do randomly. So you pick some samples from any of them at random and process them as batch 1. So in each batch, you have a representation of all the different cell types. So you process them as batch 1, batch 2, batch 3. And if plots look like this, then this simply means that the variation is driven by biology. And this is good, because then you have the same way you mix them when you are starting, you are going to see a representation of all the different cell types from different batches forming a particular cluster. But again, if you see plots that look like this, you are still having some level of batch effect, because if you see, even if you did random sampling from this step and you still end up with all the different cell types being represented here, then you are thinking, okay, all these green samples form a particular cluster. Then that means this is a batch effect still. But this is what you expect to see. And in real life representation, you can think of it this way, whereby you have T cells, NK cells, monocytes, everything just being mixed up in a UMAP. Okay, but then a good cell type is whereby you have like T cells separating uniquely compared to monocytes and all the other cell types, each of them being independent in a cluster. So the other last thing that you need to do is marker identification. And this is whereby you know particular cell types have particular genes expressed only in those cell types. For example, CD14 is a gene that is expressed primarily in antigen presenting cells. And you are going to do we are going to use our clusters to identify whether there are any unique markers that are forming our particular cluster. So from a reduced dimensional space, whereby you have these different cell clusters, you are going to make, for example, a heat map and see whether particular clusters are being driven by particular genes. Another way of doing this is you use trajectory analysis, whereby you see how cells separate. For example, if you did a Plasmodium falciparum, a different developmental stage from schizons to trophozoites to schizons to rings to trophozoites again to schizons again. So you're going to see them spread out across. And in each of this cluster on a trajectory analysis, you can identify a particular marker that is driving that particular expression. And then the other thing that you can do is you can do spatial tissue reconstruction. And this is whereby you already know which genes are expressed in a particular tissue. So you are going to separate or identify the markers for that particular tissue based on the gene that is expressed and that you already know from biology. So again, there are several tools that have been developed to do some of these steps. Dimensional reduction, whereby you are reducing the complexity of your data to very few dimensions. You can use RAT. It has several tools that can be able to do this for you. Other softwares are ScanP, Pogada, and SC6. And then for trajectory analysis, you can be able to do this with Monaco and all this, and other methods that you can use with Velocity and other softwares. So as again, you can follow this link to see all the other tools that you can be able to explore. So finding new cell types and allocating cell types along a differentiation cascade is always a big challenge. And what you want to be able to do is you want to use markers to characterize the different cell types, and also check for their differential expression, their differential transcript usage, do some level of network analysis before you can be certain that you are saying this is a monocyte is actually a monocyte. And sometimes consider some statistical tests over changes of proportion of cell identity clusters between samples, so that you can be able to be certain at what confidence level can you say these are T cells using cell proportions and not monocytes. There are several methods of doing cell annotation, and you can do this manually or automatically. So once you identify your different clusters like this, you can use markers. For example, MS for A1, I think, is a marker for B cells. Once you use it, you can be able to say, okay, this cluster 7 here is most likely B cells, because it has a high expression level of MS for A1 gene. And then you can do this over and over across all your clusters, so that you can be able to annotate them that they belong to a particular cell type. But we have quite a lot of softwares that have been developed using different languages, Python, R, that can be able to do cell type annotation. Some of them use prior knowledge, like they've been trained using deep learning methods to be able to identify cell types based on prior knowledge that MS for A1 is for B cells, CD14 is for monocytes. So it can be able to use this information to identify those cell types. So, and newer methods involve reference-based annotation, and this is whereby a reference atlas exists. For example, right now, the Human Cell Atlas, they are developing human cell atlas maps. For example, for colon cancer or healthy states. So you can use some of these cell atlases to annotate your cell types. And this is whereby you take a reference single cell sequencing data that has been annotated, bring in your query data. This is the data you've sequenced, and you map it together, and you are able to annotate your cell cluster. Sometimes you can have an unannotated cell clusters, and you can do this by using markers and gene function to identify some of those clusters. But then the good thing of using reference-based mapping is that it saves you time, because if you have 10,000 cells, or you have over 20 clusters, identifying those 20 clusters with very high confidence can take you quite a bit of time. So if you want to quickly identify your clusters, the best easy way is to use a reference single cell data to be able to classify your sequencing data. And once you do this reference-based annotation, you should use some experiments, do independent data integration, compute some level of statistics analysis, and sometimes talk to other people so that you can see whether it makes sense what you've done. So some thoughts is that, cell type, cell identity could be driven by so many things. One of it is environmental stimuli. Another one is cell development. So you could, if you sample a developing child at 1 year and 10 years, you are going to see probably not completely different, but you are going to see some level of variation between their gene expression. And this is because of cell development. Cell cycle. So this is one of the things that you should always look out for. What is the cell cycle stages of all your cell types? And you should also look at the spatial context. This is, for example, if you have cells that are being expressed primarily, for example, neurons that are being expressed primarily in the brain, you have others being differentiated in other parts of the body. There's some level of spatial context, and the gene expression levels is going to be different between the cells. So now, other things that you should consider is that there could be continuous phenotypes. For example, monocytes are always differentiating to macrophages. So this is a continuous phenotype that you should be aware of. And sometimes you have a trajectory, for example, hematopoietic stem cells that differentiate down to T lymphocytes, neutrophils, erythrocytes. So basically, at any point, if you sample this patient, at any point, you're going to see differences in gene expression because most of these cells are in a unidirectional temporal progression to their terminal state or their terminal cell type state. Sometimes you could have rare cells, rare subtypes of cells based on a particular thing that is happening in that patient. For example, if the patient is starting to develop a cancer that is unknown, or an individual and they were healthy, you could see all the healthy cells, but then sometimes you could see rare subtypes, rare cell types that you are not able to differentiate. And these are discrete types of cells that you need to pay attention to. Sometimes a spatial position. For example, you have macrophages that are always found in the gut. You have circulating macrophages. So the spatial positioning of a particular cell type can influence their gene expression profile. And this is something that you need to pay attention to. And as I said, the cell cycle of cells is also very important. Now, some thought is that you should not use marker genes, compute p-values to validate a cell identity cluster. What you basically do with a marker gene is you make an hypothesis, and you just use it to help you to annotate a particular cluster, but not necessarily identify with very high confidence. You could have confidence, but you can't use a p-value to justify that. Yeah, you are very sure that this is a particular cluster using a p-value. And there could be a lot of confounders in assigning a particular cell type. And some of these are technical variation that I've already talked about: batch effects, library qualities, cell-specific capture efficiencies, amplification bias during PCR steps, and other things such as allele intrinsic variation. These are bursts of transcription or stochastic initiation, whereby you have, because what we've learned, for example, in immunology is that you tend to have feedback loops of regulation of cells. So for example, a particular pathway, NF-kappa B, could be producing OSL gene that goes back to regulate another gene upstream, thus mitigating the expression of the whole module of cell of genes. So this could be, bursts of transcription, because, for example, if you have an infection, then you have a burst of expression. And constantly we are being exposed to pathogens in air and surfaces that we touch. So we end up having some levels of bursts of variation, especially in the immune response. Others could be allele extrinsic variation, and these are fixed cell identities, temporal progression of cells, and spatial location of cells that I've already mentioned. And these are all these things you should take them into consideration, because they could be confounders in your cell annotation or cell types. So regardless of all these steps that we've talked about that you could do, is that you should always regress out all possible technical and biological covariates, and you should normally try to do this in a joint format. So where, if you know there's a level of batch effects, allelic variation, or intrinsic or extrinsic variation, you should put all these together before you start processing your data and doing normalization, so that you can account for all of them jointly rather than do it serially. You should try and do data integration, batch correction using different methods. Also, you should be aware that some method can always overcorrect. So based on the person who developed the particular method, they might or possibly develop a method that overcorrects for a particular cell type. For example, if they use their brain tissue to develop a method and it did overcorrection, you might use the same method for PBMCs and end up overcorrecting in your data. So you should be wary of this. And the other thing that you should remember is that for each gene, the observed expression profile that is generating from single cell data can be caused by a combination of factors. And these confounders can prevent the signal of interest, the differentiated state of a particular cell, from being uncovered. For example, if you look at differentiation of naive T cell from a naive T cell all the way to a T helper type 2 cells, is that the expression levels of different genes at all these different steps or stages is that it could be different. Now, just taking this into consideration, if you add in the cell cycle state at different stages, is that and also add in possibility of apoptosis happening at any of these steps all the way to the type 2 cells, is that all these factors are going to affect your observed expression profile. So you should always bear this in mind, that there is a potential that the expression profile you are seeing could be not because of your clinical phenotype, but basically because of the cell being under differentiation, cell cycle, or apoptotic. You could have collected your samples from terminal cancer patients and your cell types from healthy patients, and you see that the expression profile for a particular cell type is not significantly different. It's because there could be just cells that are not involved in the cancer, but are just changing as per their differentiation state, the cell cycle, and apoptotic step. Now, finally, let me briefly talk about differential expression and how you determine differentially expressed genes. And this is what I would say most people want to do a single cell analysis to be able to see and say is that particular genes are expressed highly in monocytes in patients with cancer compared to healthy people. So this is more like the terminal step of single cell analysis. So we often do not have, defined set of experimental conditions. And what we do is that you end up doing pairwise comparisons of gene expression between pairs of cell clusters or pairs of groups of individuals. So, but it always goes down to cell clusters between two different phenotypes. And normally people use Wilcoxon's rank sum test as a default statistical measure to do this. Other methods include Poisson, Nigbon, and others, and so many others that you might encounter. So, but once you do your differential gene expression, one of the things that you can do is that you can correct for other confounders that you are not able to adjust for during the pre-processing steps. And you do this by taking the confounders, by evaluating the confounders, and if possible, put them in your analysis as a regression, in a regression, in your regression equation. So for example, you have your cell type matrix and you have some levels of biological variability, cell cycle, technical variation. You can take all this, if you're able to put them in a format that they can go into a statistical equation, you can put all of them in, for example, in a zero inflated model, whereby you take your gene expression matrix with, dimensional reduction or your cell clusters, and you add in all the biological factors plus known parameters of the biological factors. So the non-biological factors and unknown biological factors. And also you add in your regression model the observed technical covariates plus all the unknown parameters, and you add in all the hidden technical covariates that you think of plus their unknown parameters, and the zero mean noise across the data set. So once you do this, you end up with a model that is going to be able to adjust for all the noise that might be in your data that you are not able to remove during pre-processing. And at the end of the day, you're going to have a differential expression that you can be able to trust, because its expression levels is not going to be within the noise section, or is not going to be in a log normal expression, but then you are going to have a zero inflated expression, log expression, and a normally distributed expression levels for your genes. So some of the things that you should consider is that differential expression testing should not be performed on corrected data. So normally on when you are doing differential expression, you use your raw gene counts. And sometimes you do this on the raw RNA gene counts, not on the batch corrected data. But if you've done batch correction and denoising, you can do your differential expression on this. But instead, you should use all the data that has the technical and biological covariates that you can be able to remove them within the model. Sometimes do not rely on differential expression testing tools to correct models with confounded covariates, because sometimes some of the softwares are not, I'll use the word, aware of the covariates that might be existing within your data set. So you should be careful when you're doing differential expression testing. Some of the recommended methods are MAST or LIMA for differential expression testing. Sometimes you should be wary of uncertainty of, regulatory relationships within your gene sets, and modules of genes that are enriched for regulatory relationships might be more reliable than individual ages. This is because if you know, for example, you have high expression of NF-kappa P target module, then because of a particular infection, and this has been tested previously in biology. So once you start seeing that the expression levels of that particular target module in the presence of a particular disease is not what is expected, then you should be wary of that and try and adjust that in your model or in your regression analysis. Now, I would like to summarize that, all I've talked about are things that are biological, but you tend to consider them in your computational analysis. And it's very important that you pay attention to the things that you need to correct for during your analysis than the things that you need to move through smoothly. Because most of these things are being containerized into pipelines that you just do, a few commands in R or Python, you are able to get your results. So, but at each stage of your analysis, take it with a pinch of salt and look for possibility of some level of technical variation or technical covariates, biological covariates that you can go back within your data set and correct for. So it's a call to all of us that we need to contribute to the Human Cell Atlas. And this is because there are already existing tons and tons of data sets from different cell types, but we haven't covered the entire human cells. So your new data that you're going to generate from your cell type, from your experiments or your research, is that you can put that new data and add it to the existing cell type so that we can, at the end of the day, we'll have very well-defined cell type ontology for the entire human cell, all the human cells. And this is a vision that we hope, or we look forward to achieving with the Human Cell Atlas. So thank you very much.
Thank you, Collins. That was an excellent presentation. Does anyone have any questions for Collins? Feel free to either just raise your hand, take yourself off mute and ask the question, or you may also type it in the chat. We'll give it a couple of moments here for questions to come in for Collins. So Christo, I see your face. Do you have
yes, please. Are you hearing me? Actually, I do not know.
Yes, if you have a question, please ask it.
Yes, sure. Collins, thank you so much for this very fine talk. I was wondering, how do you evaluate the influence of the spatial disposition of cells, for example, in a tissue? I mean, how to evaluate if they cluster among themselves or probably cluster with another cell type in a significant manner? Thank you.
Yeah, so I think there are methods that have been developed for spatial deposition of cells, but what you need to do is use marker genes. So if you already have like prior knowledge about a particular marker that is used to map particular cells in a particular. So for example, you take a whole tissue and you have, you have different cell types segmented across the entire tissue, and then each of them should be expressing a particular marker type, a marker of interest. So you take those markers and you try to see that there is no overlap in the expression of those markers in different clusters of your entire tissue. And this is the other thing is that you could use, for example, histology to confirm that you're having very good spatial distribution of your cell types. So I don't know whether I've answered your question.
Sure you did. Thank you.
Yeah.
Thank you, Collins. Did anyone else have a question for Collins? Okay, so if there are no further questions, thank you, Collins. I appreciate that. We do have a break, it looks like. Let me just see what time we have here. Right, so we're running right on time. So what we can do is we can plan to take a 17-minute break and come back in 17 minutes. So that will be 2:45 p.m. GMT, 9:45 a.m. Eastern time. So go stretch your legs, get something to eat, and we will be back here with Vincent, who will prep us with our studio. So see you all soon. Thanks, Vincent, and thank you, Collins. To you.
Okay, so let me quickly share my screen now. I think I'll get one of the accounts here first.
So we can't see your screen if that's what you were asking, Vincent.
Let me just share again. I'm wondering why it's not appearing. Let me check again.
Sure.
Can you see now?
Yes, I can see that. Great.
Okay, yeah, so let me quickly work on that. Okay, so let me also share the link that we are supposed to use. So for those who have received the details, account details, you have to visit the site. I am putting it here. It's a web-based platform. So if you get there, you have the username and password. So you are supposed to enter your username and password. So I will try with a dummy account now so that you see what I'm talking about. I think I should have one of the accounts here.
And just so everyone knows, this, your username and password were sent to you last week. The header on that email was called Important HCA Africa Introduction to Single Cell RNA-seq Analysis Workshop Pre-Work. So you should check that email for your username and password. If you have issues accessing that email, you can either let me or Tracy or Elliot know. You can chat us and then we can send you your username and password.
Okay. So, okay, yeah, so I think everything is ready now, so I'll proceed. So I'll use this account. So the account you received the username will be something like the SCW, and then you add your number. So let me use this one. This SCW listener. So I have to enter my password, and then I will log in. So in case you have any issues with logging in, you can still contact us and I will resolve the issue for you. So after you have logged in successfully, you have your R Studio being displayed for you. So here we have the R environments. You can write your codes. You can also check the data you have in your account. Now, you should have all these files in your account. We'll use them in this course. So if you are here, you can also, there's a terminal you can use. You can create a new R script if you want to, and then just type in your comments, or you can use the already existing R scripts here, which you can go to file, you can go to open file, and then you can select the ones here. Okay, the instructions that will be given will tell you which of the R scripts you should use. So those ones, you can open them and then you can run them as you see, and you can also include your own commands. Let's say print one, and then you can execute it. So it's basically so we have all the packages installed, so you don't need to worry about installing any packages for this particular exercise. So you can use it, you can go all out to use that, and then get your hands dirty with R. If you also want to download, you can come here. Here you can upload and you can also download when you are in this section. So you can try and then get everything that you need here. So that's it. We also made a dataset available on GitHub. So the link will be shared to you so that after the workshop, if you want to try it on your own laptops, you can still do that without using the server. So there's a summary. I think Sarah will go into details with the tutorials itself, but it's important you set it up now so that she can proceed with the rest of the activities. So any question up to now? You can also type it in the chat box and then we will get back to you.
So just to clarify, Vincent, all of the participants should be logging into our studio using their username and password. Is that correct?
Yes, yes.
Okay, so please, everyone, do that. If you have issues or if you need a reminder of what your username and password is, please let either Tracy, Elliot, or me know, and we can send that to you. So we'll give everyone a couple of moments to do that.
Okay, so I think I've seen some people logging in already. Yeah, so I think, yeah.
Great. So maybe we'll give it a couple of moments until we move on, but everyone here, please do start logging on as instructed. So Vincent, there's an issue with Jelta saying that the login screen is rebuffering, meaning I can't actually get into the RStudio page. Is that normal, or any advice there?
Which web browser are you using? It should work on
I'm using Chrome.
Pardon?
Chrome, Google Chrome.
Yeah, Chrome should work fine, but if not, try then switch to Firefox and then see.
Okay, I will have a go.
Okay, yeah, so try and then let's see what you have. Okay, yeah, so someone is asking about the post login. Yeah, so the post login, So after you log in, this is what you will probably see. You have this blank screen and you have the files here. So the files here, you should see all of them because it's a copy for each one. So you will use all the files here for the exercise. So make sure you have all the files that you see here. If you don't have them, prompt me so that I make a copy into your respective accounts. So if you are here, it's an R interface. So you can type your R, maybe print hello world. You can type your R commands. You can also load R scripts. So I was making a reference to how you can load these files here. So you can go to file, and then you can go to open file, and then you can open each of these files. But for now, I'll advise you not to modify the scripts because we will use them later, but just try and then see how you can open that. So you can click and then you can open it. But then Sarah will take you through the R basics. So you don't have to use this one. This one will be used after you've gone through the R basics, but I'm only showing you how you can access them. You can try with new scripts. That one, you can enter your own commands. So I think this one will be good for the basics. So you can get a new script like this, and then you can type your R commands. You can also use the console here, and then type your R commands if you want to do that also. So there are different options for you to use the R environment, and there are lots of packages you can use for your exercise. So you don't have to worry about the packages as well. And then on this side, you can upload, you can also export files. When you come to export, you can export files that you have created on in your accounts to your local PC. That's also possible. And then basically that's what you see. But in case you load any data sets and you are working, you will also see some information here, which is to tell you that you have some data sets in memory and you are using them. So basically, this is how the course is going to be like when we use the R interface. Okay, let's check. Okay, issue has been resolved. So we are okay now. Anybody else having issues? Okay, some are saying they don't have their files. Those who don't have the files, you can just send me your user account name, not the password, just the user accounts. I will try and then check for you what is happening. If you receive a notification with the username and password, then you should have the files in your accounts. So if you don't have it, send me your username. I will check and then investigate why you don't have the files, and I will copy them for you. Okay, so I assume that everyone has been able to log in successfully. So I think we can proceed. Yes, everyone has the files in their respective account. So if you receive the information, you should have the files in your account. You should see them. If you don't see them, try to refresh your browser and then you see the files. Has everyone seen the files in their respective accounts after opening the R server? Okay, I think everyone has logged in now successfully, so we can proceed with the rest of the exercise. But I'll still be available. If you have any issues, still contact us and I will check for you. So I think we can go on.
Sorry, Vincent, could you please send the link again?
Okay,
to us to do?
Okay, let me. send the link again.
Do you know why I wanted to make a feature?
So I've sent the link again. So try and then log in. I think let's give them 5 more minutes, and after that, we can go to the next part of the work. Okay, I think everyone is set up now, so we can proceed. Okay, so I think Sarah can take over now.
Great, so Sarah, are you there?
I'm here.
Great, so let's move on to your presentation.
Sure. Let me share my screen. Can you guys see my screen?
I can.
Okay, great. Thank you. Hi, everyone. My name is Sarah Ingabire. I am a research technician from the Shalek lab, and I am very excited to be part of this workshop. So today we're going to talk about the introduction to R. So we'll be learning about some basic concepts and notions about like how R work and the thought process around it. So if you have any question, please feel free to stop me, or you can raise your hand, or like you can send your question in the chat, and I'll try to keep a look on that. Okay, so let's get started. So first, what is R? So R was created in the 1990s by statisticians Ross Ihaka and Robert Gentleman. It is a comprehensive programming language which is used for statistical analysis and graphics. It is also considered as a dialect of the S language, meaning that it simply means that R was built on the same basis and execute the same function as the S language. So to give a little bit more background, the S language was developed around 1976 at Bell Laboratories. And one of the key limitations of the S language was that it was only available in a commercial package, which is S+. So when these statisticians were developing R, they were convinced to use the GNU, which is the General public license, to make R a free software. And this was critical, and because the entire R system became accessible to anyone who wanted to use it. So you can obtain R in several forms. You can either obtain it from the sources if you use Unix and Linux machines, or you can also obtain it from some precompiled binaries if you use Windows, Linux, and Macintosh. So next, let's talk about why R. So why should we care about using R? So like I said, R is a free software. Anyone can have access to it. It's a free and open source. It is also one of the most widely used software environments in data science. It has an effective data handling and storage facility. And one of the key features for using R is that it also has many functions. And these functions can provide a large variety of statistical analysis and graphical facilities for data analysis. So these functions can also be displayed immediately in their own window, and you can also save them in various formats. So you can save them in JPG, PNG, or PDF, etc. Another key feature for using R is that it is actually possible to combine these different statistical functions. So you can combine them together in a single program, and that would allow us to perform more complex analysis. So now let's go through the steps on how to install R. So there exist three basic steps on how to install R. So first, we need to install R, and then next, we need to install R Studio, and then finally, we need to install R packages. So at this point, you may question yourself, what is the difference between R and R Studio? So R simply is a program that runs all your code. And then R Studio is another program that's going to allow you to control R in a more friendly way. So it has these cool features, like it has a powerful text editor. So this is where you're going to write all your code, as we'll see later. And then it also has another place called console. So console is basically where you're going to run all the codes that you've written in the text editor. Okay, so first, let's talk about installing R. So we use the CRAN website. So CRAN simply stands for the Comprehensive R Archive Network. And it basically stores all the files that are needed to install R, and gives you all the instructions that you need. So, so if you have Linux, you will click on this one. If you have Mac, you'll click on this tab right here. Or like if you have Windows, you'll click on this download R. So let's go through a couple of them just to kind of have an idea on how to install R on your computer. So for people who use Mac, you can either scan this QR code right here, or like you can open this following website right here, and this should take you to a page that looks like this, and it shows basically the latest version of the PKG file, which is what we need. And you can go ahead and click on each one of them depending on the type of Mac that you have. So if you have Intel Mac, you'll click on this. If you have M1 Mac, you'll click on this. And once you click on it, those files will be able to download themselves on your computer. And then if you open the downloaded PKG file and then you open R will be good to go for people who use Mac. So for people who use Windows, you can go ahead and scan this QR code right here, or like you can open this following website, and this should take you to this page right here. And all you need to do is to click on this tab, which is the latest version of R. And when you click on it, you'll need to double click on the R installer for it to launch. And then when it launches itself, it will ask you to choose your choice of language. So when you choose it, you click OK, and then it's gonna prompt you to read the important information of the license. When you're done reading, you click next. And then you will want to choose the path, like the destination location to where you want your folder to be. And then once you choose it, you click next. And then it's gonna prompt you to select the components that should be installed for R. So ideally, we want to keep all the components checked. And then you're gonna click next again. And it's gonna want you to start to customize the startup options. So you can go ahead and accept the defaults ideally, and then you click no. And then once you're done, you click next. And then it will give you the option to select a start menu folder, but by default, you won't have access to this tab right here. So you will need to uncheck this box here. And then if you want to create a start menu folder, and then you're gonna click next again. And it's gonna give you additional tasks. So you can either create a desktop shortcut, or you can create a quick launch shortcut. So once you choose, you can click next again, and then R will start to get installed on your computer for people who use Windows, and it will also be good to go. So now let's talk about installing RStudio. So to install RStudio, the process is a little bit similar. So you can go ahead and scan this QR code right here, or you can open the following website. And then this will bring you to this page right here. So you can either select RStudio Desktop or RStudio Server, which are free. So both of these are similar. The only difference is that one has access via web browser, the other doesn't. So go ahead and choose whichever is most convenient for you. And then, so for people who use Mac, it's pretty straightforward. You will click on this download RStudio for Mac, and then it will start downloading itself. And then once it's done downloading, it will be good to go. But let's go, for example, for people who use Windows. So you will click on this. So this should be like the last version of RStudio as of now. And then when you click on it, it's gonna give you it's gonna guide you through the installation of RStudio. And then all you need to do is click next, and then you will choose the destination folder as we've seen for R. And then once you choose the destination location, you click next again. And then you also have the option to select the start menu folder. So you can go ahead and enter the name of RStudio, like if you want, and then you're gonna click install, and then RStudio will start installing itself on your computer, and will be good to go. So now let's talk about installing packages in R. So like I said, R is a free software and it comes with a built-in set of packages. And packages can be seen as a form of bundle or a collection of functions, and they can be created by anyone. So here is a list of packages that are most commonly used, at least especially for beginners. So we're gonna go through each one of them just to kind of have an idea of what do they mean, like why do we use them, and have a sense when we're loading them, just have a sense of what they actually mean. So here we have ggplot2. So ggplot2 is pretty popular for data visualization. So it allows you to graph most kinds of data in a very simple way. It also has some useful commands that help you plot some complex plots. Here we have patchwork. So patchwork is pretty popular for data manipulation. So it makes the plot composition in R extremely simple and powerful. And then we have dplyr. So dplyr is also, we use it for data manipulation as well. So it makes the tabular data manipulation fast and easier. And then here we have tidyr. So tidyr makes it easier to tidy your data. And then to tidy simply means to organize. Another way to think about it, like tidyr has three main functions, which is separate, gather, and spread. So if you, for example, if you have like two variables clumped together in a single column, you can tease them apart using the function separate. And then here we have string R. So strings are a bunch of characters. So string R provides a set of functions that are useful for data cleaning. So if you want to change a case of the word in your code, string R is really good for it. And then we have Seurat. So Seurat is designed for quality control, analysis, and exploration of single-cell RNA-seq data. So it helps you identify and interpret the sources of heterogeneity from the single-cell transcriptomics measurements. And then here we have matrix. So Matrix collects data sets and arrange them in two-dimensional rectangular organizations. Okay, so now let's talk about how to install packages in R. So there are various ways of installing packages in R. You can either use the CRAN, which is the Comprehensive R Archive Network. So this is the main repository for software packages. It holds over 18,000 packages. Or you can also install it using Bioconductor. So Bioconductor is most commonly used in computational biology and bioinformatics, and it holds over 1,800 packages. You can also get the packages in R downloaded directly from the GitHub Online software, where you're gonna use the DevTools package. Or you can also obtain them from the source package, and all you need to do is open the terminal app, navigate to the directory where you currently have the file, and then you execute, usually like you use this code right here. So you enter the package name and then you do .tar.gz. So this will be how it would look like if you run it in the terminal app. Okay. So now let's get started with RStudio. So RStudio, like Vincent was mentioning, it comes with different compartments. So here we have the text editor that I was talking about. So it kind of works as Notepad or text edit. So this is where you're gonna write all your codes right here. And then we have the R console. So the R console is basically where R is waiting for you to tell it what to do, okay? So this is where you're gonna run all the codes that you've been writing in the text editor. And then here we have the environment and history tab. So the environment shows all the variables that you've been generating during the course of programming. And the history tab is gonna show you all the commands that you've been running. during the course of programming as well. And it's temporarily saved in our workspace. And then here we have this compartment has different tabs. So we have the files, the plots, the packages, the help, viewer. So files is gonna show you all the files and directories within the R workspace. The plots is gonna show you all the plots that you've been generating during the course of programming. Packages show you a list of packages that you've installed so far. It can also give you an interface of the new packages that you need to install. And the help is really useful and important because this is R comes with a built-in help system using R documentation. So whenever you need help how a function works, if you're not sure about it, this is where you're gonna go to get help from. And it's gonna give you like a description about the function that you wanna use. It shows you how to use it. It also shows you like what do these arguments mean and how to use them. So it's really, really useful. Okay, so let's get started with how do we create and delete objects from memory. So R directly executes commands without requiring to build a complete program. And if you want to test a code, you simply go to console. And here, for example, I wanted to know what is 1 + 2. And then for it, in order to run your code, you need to hit command and enter. And then once you hit that, you get the results. But like, if you want your results to be saved, you need to assign the value to a variable. And to do that, we use the assignment operator. So you can either use this arrow right here, or you can use the equal character. And then, so here, for example, like this is what I would write instead in the console if I want it to be saved. So I named my variable to be named n, and then I assigned, this is the value that I wanted. And then when you hit command enter, you will see that in the environment, we have the variable that I called n, and then it's equal to the value of 3. So you can also delete objects in memory, and we simply use the function rm, which simply stands for remove. So you can either delete one object or multiple objects. Okay. However, there are a few things to keep in mind. So R is case sensitive, meaning that lower cases and capital cases are going to be treated as two different objects. So if you write numbers like this, and then you write and again numbers like this with a capital letter, R is going to treat, although they would have the same value, R is going to treat them as two different objects. So those are the kind of things that you need to pay attention to. And in R, space does not matter. And also the object names cannot contain strange symbols such as plus, minus, exclamation point, or a pound. However, you can use the dot or underscore. Also, the object's names in R can contain a number, but they cannot start with a number. Okay, so let's talk about the R help that I was talking about. So R comes with a comprehensive built-in help system. So the online help tool gives very useful information on how to use the functions. And you can simply type in the R console to get help. You can simply do question mark and then the name of the function. Or you can use the function, you can do help and then a parenthesis and then write in quotation mark the name of the function, or you can just simply write help parenthesis the name of the function, and this should display a page that looks like this. When you run this, you shall see like in the help tab something that looks like this. So it gives you a description, the usage methods, and it also gives you like, so it gives you how to use it. So here, like you see X, and then if you go to arguments, it'll show you like, oh, what is X? So X is an R object, and this is actually this is how actually you need to think about it. Okay, so now let's talk about reading data in R. So R is capable of reading data from most formats, including files created in other statistical packages. So if you're not sure of your working directory, you can simply use the command getwd parenthesis to find the directory. So if you hit command enter, it will show you the directory that you're in. And then if you want to set working directory, this is how you will do it. So if you once you find the get working directory, you can simply copy and paste it right here. And then that will prompt you to set it as a working directory. Okay, so let's suppose you want to load the Titanic dataset. So if the dataset is an existing R package, you can load the package using the function data, and then you write the name of the dataset that you want. This is in the case that you want to see what's the dataset about. You can also change it to .r data format. So all you need to do is load Titanic, and then R can also transform files, TXT file or other text formats. All you need to do is use this function read.table and then parenthesis, the name of the data set, and then it has to be .txt. It can also transform files like Excel files into CSV formats, and then all you need to use is read.csv and then do Titanic. This is supposed to be CSV, sorry. And then for data formats, you can load the foreign library using read.data function, and then so this should look like this when you run it. And then to save objects into these formats, you can simply use these commands like write.table or like write.csv, etc., as we'll see in the lab. It'll make more sense. Okay, so now let's talk about the R objects. So there exist different types of data types. So we have numeric data type, we have integers, we have logical data type, and we have character data type. So numeric data type is for numerical values. So this can simply be like any numeric values. Like it can be positive, it can be negative values, it can be decimal points, it can be anything. Okay, and so here's an example to test it. So I assigned my variable to be named num, and then I want num to be equal to a value of 1. So to check the class of my numeric value, I simply do class, and then I put in the variable that I assigned it to, which is num in this case. And then when you hit command enter, it's gonna show you that, oh, this is a numeric data type. And if you want to check, like, is this a numeric value, we simply use is.numeric, and then parenthesis, you put in the name of the variable that you assigned it to, and then it will be true. And then if it's not numeric, it will be false in this case. So we also use as.numerical function if you want to convert the values of other data types into numerical values. So if you look in the environment, you'll see that the num that I assigned has a numeric data type. It has 1 element, and the num is equal to a value of 1. Okay, now let's talk about the integers. So the integer data is type is used for integer values. So these are like positive values. So in this case, I chose my x to be equal to 20, and then I want to check if this is this an integer? So since integers are also part of numeric values, because like, remember, numeric values are also positive values. So you need, by default, this will be numeric data type, and you need to transform it into, if you want it to be an integer, you need to specify as such. And to do that, we use the function as.integer, and then you put in the variable that you you want to change the integer to, and then if you check the class of the variable that you assigned, you'll see that this is an integer. And then if you go to the environments, you'll see that I've named X, and then it has an integer data type, it has one element, and this is the value. So every time you create an integer in the value, like you'll see the value, and then you'll see this letter L. So this shows that it's an integer. Okay. So now let's talk about logical data type. So these are simply the Boolean truth values. So logical, like it can be true or false. So here is, for example, here I assigned my variable to be named Y, and I want my variable to be true. So if I check the class of Y, you'll see that this is logical. And I want to ask, is this a logical data type? So I simply use the same function, is.logical, parenthesis, the name of the variable, and this will be true. And if I check in the output environments, you'll see that Y is saved as a logical data type. It has one element, and it has the value of true. Okay, so character data types are simply they store like character values or strings. So it's anything that is in quotation marks. Like here, for example, so as long as it's in quotation marks, it makes it a character. So here, for example, I named my variable to be Z, and the value of Z is I love single cell. So this is going to be treated as a character. And if you check the class of Z, you'll see that this is a character, and you can also do the is.character Z, like we've seen like others, and this will show you that this is true, like it's a character. And then if you go in the output environments, you'll see that Z is saved as a character data type. It has one element, and like the elements here will be I love single cell. So there are a few things that you have to keep in mind here, like you cannot, for example, if you put quotation marks inside of quotation marks, this will be displayed as an error, like R will not run it. So if you want to put like quotation marks inside of these quotation marks, you'll need to put a backslash before just to tell R, or this is a special character and you shouldn't consider it as a quotation mark, and it will let it run. Okay, so now let's talk about how we can manipulate those data types that we just seen in R. So we can do it such as like using factors, vectors, lists, data frames, or matrices. So we're gonna go through each one of them and see how they work. So factors are seen as categorical variables, and they store both string and integer data values as levels. So here, for example, I chose my level to be, I chose my categorical variable to be sex. So there are males and females. So in this case, I chose the sex categorical variable. And then to create a factor, we use the function as.factor to precise that I want my data type to be a factor. So I assigned it to be named my factor, and then I used the function as.factor, and then you open the parenthesis, and then you write in the categorical variables. So this is treated as a factor of one level. So we'll see later how it works when you put a bunch of categorical variables together, like the way it varies with the factor of level. Okay, so vectors are homogeneous, and they are basically elements of the same data type. And in order to create a vector, we use the function C which simply stands for concatenate, and concatenate simply means putting things together. So And if you want to check the class, you simply use the class function right here, and then you put in the name of the vector to check if it's like what type of vector is it. So like we've seen numerical data type, character data type, factors, logical data type. So we can actually put them, like put a bunch of characters together. We can put a bunch of numerics together. We can put a bunch of logical together. So when we're doing that, we're creating the data type called vector. So for example, here, if we put a bunch of numerical values together, we're creating a vector. And to create a vector, you simply use this function C right here. So I first assign the name. So I want my numeric vector to be named numeric vector. And then I use the function C, and then these are the values that I want. And then if you want to create character vectors, remember characters are like, you basically need to quote them to make it a character. So here is a list of characters, and characters can be anything. They can be letters, they can be numbers, they can be a word, they can be like a phrase. So they can be anything, right? So as long as you quote them to make sure that to tell R, oh, so this is a character. And then to make it a character vector, you simply add the function C right here, which stands for concatenate. So you're putting a bunch of characters together, and you're creating a character vector. So this applies the same thing for the factor vectors. So the factor vectors is kind of similar as writing a vector of character, but the only difference is you need to give it the as.factors function to make sure that this is a factor. Also, like, remember, factors are seen as categorical variables. So in this case, remember how first I chose a sex as a category. Here, my category of choice are letters, are alphabets, right? So this will be a factor of 5 levels because it has 5 different letters. Okay. And then it's a vector because we use the function C right here. And then it's a factor vector because we used this function as.factor. So you basically treat it as character vector, and then you give everything like the as.factor function. So the same apply for logical vectors. So all you need to do is to put a bunch of logical variables to, like, you put them together, and then when you do that, you give them the function C to make it a logical vector, and then you'll be creating a logical vector. Yeah. So, however, there are certain rules that apply if you decide to put different data types together in one vector. So remember, vectors are homogeneous. It has to be elements of the same data type. So if you apply like different data types in one vector, it will eventually be converted into another data type. So if you choose to to combine logical values with numerical values, this will automatically be converted to numerical. So it will be treated as a numeric vector. And if you choose to combine logical data type with numerical and character data type, all of this is going to be considered as a character. So everything will be considered as a character vector. So here's an example. So for example, if you choose to create a vector that contains different data types, so in this case, 1 corresponds to numeric data type, and then false correspond to logical data type. So if you put them together, this will be converted to a numerical vector. It will be saved as a numerical vector. And then if you combine logical, numeric, so like this is numeric, this is logical, this is a character because it has quotation here, this whole thing is going to be converted to character vectors. So those are the things that you need to keep in mind if you apply different data types, if you put them together in one vector. Okay, so list is another data type that is used for data manipulation. So list is non-homogeneous. So vectors are homogeneous, but list is non-homogeneous. So list can contain anything. It can contain numbers, it can contain characters, it can contain another list inside of a list. You can put in a matrix, you can put in another function inside of it. So list can basically contain anything. And to create a list, you use the list parenthesis function. So here I assigned my variable to be called my list, and then I use the list function to create a list. And then this list contains, you can put in numeric vector, you can put in logical vector, you can put in character vector, you can put in number. You can actually put another list inside of this list. You can, and then I added also a function. So we'll also go through it in the lab, and you'll see how when we run it, the way it would look like. Okay. And then next, data frame. So data frame is basically a list of equal length vectors. So each element can be thought as a column, and the length of each element of the list is the number of rows. So one thing that you need to keep in mind when you're creating a data frame is that the vectors have to be equal length. Like they have to, like, for example, this is numeric vector, okay? Like I defined my numeric vector to be this, right? So I used the function C to create a numeric vector, and then these are the numerical values inside of it. And then if you look at this contains 5 elements. And I also created this logical vector, and then this logical vector as well contain 5 elements. So there is false, so 1, 2, 3, 4, 5. And then this also is 1, 2, 3, 4, 5. So they have the same equal length, both of them, right? So that means you are able to create a data frame. If it was in the case where like the length was not equal, like this will run into an error, okay? So I created my data frame, so I named it my data frame, and then to create a data frame, you use the function data.frame. And then I put in the numeric vector that I assigned here, and then I put in the logical vector that I assigned here. And then, so you can also name this. So like I assigned my numeric vector to be called X, and then the logical vector to be called Y. Okay, so this is a matrix. So matrix is another data type. So it's a combination of 2-dimensional objects with the same data type. And to create a matrix, we use the function matrix. And matrix can contain like one element, like one data type, right? Like, so here, for example, like I created my data type, I chose a character vector. So I used the function C to create a vector, and these are my character values, okay? Because they are in quotation marks. So I can now create a matrix, and then to create a matrix, so you need the data type, and then you need to define the number of rows and the number of columns. And if you want to check the class of which class is it, you can simply do class to use the class function and then put in the name that you named it. So in this case, my matrix. And then when you run it, you'll see that this will be called, this will be like basically matrix data type. Okay, so to conclude, we've went over different data types. So we've talked about the numeric integer data type. We've talked about logical data type. We've talked about the character data type, as well as the factors. So when you put a bunch of numeric values together, when you put a bunch of logical values or character or factors, you're basically creating a vector. You're basically creating a data type called vectors. Okay, and vectors can be so like a data frame is basically a list of vectors of equal length. Okay, so like they need to be equal length in order to create a data frame. And then here we have a matrix. So matrix is basically a combination of two-dimensional objects with the same data type, and all elements have to be from the same data type. So vectors are homogeneous, okay? Data frame, you have to create them, they have to be like a list of those vectors, and they have to be they have to have equal length. And then for matrix, it has to be the same data type. So you choose a certain vector that you've created, and then you define the number of rows and the number of columns. And then list can be anything. So list can be a data frame, list can be a vector, you can add a matrix, you can add a list inside of a list. It can be these numeric, logical characters or factors. So lists are non-homogeneous. Yeah, so thank you for listening to me. And are there any questions?
Thank you, Sarah.
Hi.
So any questions for Sarah? Feel free to either just unmute yourself and ask her directly, or you're welcome to type your questions in the chat box. So I see Danielle Nagy. Do you have a question for Sarah?
Yes, thank you very much. Can you hear me? Oh, cool.
We can hear you.
For the presentation. I have a quick question. I've already downloaded RStudio on my computer, and I was wondering that from the browser version that we got the email, we got the password in the email. I was able to, so I found the console and script editor and everything else, but I was wondering how I can save whatever I wrote in the script editor to my computer. So if I go to File and Save As, then it might be just me, but I can't find an option to put it in my computer. It feels like, it seems like it wants to save it online, but I would like to access it offline. How's that possible?
Yeah, sure. So tomorrow in the morning, we'll be going through the lab. So I'll be taking you guys through all the steps, like if you want to save it to your computer so that you can practice later, or if you want to edit everything about it, like we'll cover everything tomorrow in the lab, and it will make more sense.
Sorry,
Yeah, of course. Yeah. So this was just a theory, like the theoretical part, just to kind of get an idea before actually jumping into RStudio, like the lab.
Thank you very much.
Yeah, of course.
Any other questions for Sarah? Any questions? All right, any questions before we depart today, either housekeeping or operational questions? Feel free to ask them now. I do believe that this was the last talk of today, if I'm not mistaken. Is that right, Sarah? Okay.
Yes. Yeah, that's right.
Okay, so maybe I see that Or is online, or you want to take yourself, there you go. Hey, Or, any housekeeping or things that you would like to address before we get into tomorrow that people should be looking out for?
Nope. I think tomorrow should be fun, getting to do a little more coding.
Great. Okay, so we will begin tomorrow, same time as we did today, 7 a.m. Eastern time, 12 p.m. GMT. Tomorrow we will be going into breakout sessions where you will do more of the practical hands-on work. So please join us, as those breakout sessions are not recorded, and that's kind of where you get to test and really put the knowledge that you're gaining to a test, I would say. And I do see somebody asked a question, just trying to find that. We will be able to receive the, yes, so recordings from any of the talks will be posted on the event site. So you'll just want to log on to the event site, And they'll be posted, I would say, Tracy, what do you think, in the next, here, probably 2 to 3 hours. Takes a little bit of time, but you can feel free to check back in. And I also believe that our speakers, so Sarah, Collins, Daniela, they'll be sharing their slides in the resources channel, on the Slack resources channel. So check there too. They are excellent trainers, and I am sure they will get back to you as quickly as they can if you have questions after reviewing the material again on your own. So Tracy, Elliot, any administrative housekeeping things you want to talk about?
No.
Are we okay?
Thank you, Christine. I'm fine. Thank you.
Excellent. So I want to give a special thanks to Sarah, Collins, Daniela, Vincent, and then Alex Shalek, who is not with us anymore. He's with us, but not here with us. But thank them all for their time and energy for today's presentations. And we will look forward to seeing you all tomorrow. So thank you, thank you. Bye, everyone. Bye.
Bye, everyone.